still using that photo, Katie?
I am. It's a great photo. Oops. Okay, so we are live. So before we begin, I'm just going to start with our land acknowledgement and acknowledging that the land that we're on is the traditional territory of the Three Fires Confederacy of the First Nations, comprised of the Ojibwe, the Odawa, and the Potawatomi. We respect our longstanding relationships with these First Nations people in Windsor, Essex. And as we like to do at the beginnings of our meeting, in the spirit of what we've learned from our Indigenous educators, is that we'll start with a statement of gratitude and a statement of hope. And today I'm very, very grateful that we've got these really awesome presenters here to help us understand harmful algal blooms in the Windsor Essex region. And I'm hopeful that we'll have a really great meeting and we'll all learn something today. So thank you for being here. So
Thank you for that, Katie. So with that, I'll call the meeting to order and welcome you all here. Now we have, I make it to be 11 members present, so we have a quorum.
Perfect.
Sam will keep track of who's attending. Jenny, let's see, chair is welcome. Okay, so if I can find it. It's really hard to keep this up on the screen. So I want to welcome you all. We have several members, some members absent, some by car phone, right, John? Or
I'm here.
And we have 3 presenters with us this afternoon. We have Chelsea Salter, master's student at the University of Windsor, my old stomping grounds. Emily Varga, also a PhD student from the university, and Kyle Davis, who's a water compliance coordinator for Lakeshore. And those 3 individuals are going to be presenting today. We thank them for coming and look forward to hearing from them. We have a new director at the province for the Source Protection Branch, Kirsten Corrigal. I'm going to let Olga make some comments about her. And I'll meet her one-on-one on virtually one-on-one in the end of November. I have a meeting set up with her. We did have a chairs meeting in September and mostly talked about those areas not yet covered by source protection plans as places that are on wells or community wells or other than municipal water systems. And when we first in 2007 started talking about source protection plans, it was understood that it was only municipal plans that we were dealing with first, but there is some feeling amongst the chairs that other kinds of water systems should have the same measure of protection if we can get to that, get the province to take that under their wing. We have a new SPC member in waiting, Chad Quinlan, who's going to be our second ag rep, replacing John Mark Beneteau. John Mark has got such a full plate that he has difficulty getting to the meetings that everybody else can. So Chad is going to take over once the Source Protection Authority appoints him. And with that, I need to be able to see consensus. So are there any declarations of conflict of interest? And I see a whole bunch of not shaking heads. Nobody put their hands up. Okay. Seeing none, the agenda was pre-circulated. A motion to approve that agenda by Matthew, supported by Tim. All in, any addition, deletion, correction? All in favor? Opposed, carried. Thank you. I skipped by, that was the agenda. The minutes of our meeting on March 10th need to be approved. They were pre-circulated as well. Motion on those minutes. Nelson and Ron. Any addition, deletion, correction? All in favor? Opposed, carried. It's working pretty good. There is no correspondence. So we're at number 8, which is Olga Udina. Welcome, Olga. And it said you have an update for us.
Thank you, Chair Firth. Can you guys hear me okay? Perfect. Yep. Thanks. And I don't have many updates for you today, but maybe start with what Tom was mentioning about some changes in leadership that we've had at both the branch level and also at the ministry level over the summer. So we have the new director who joined us at the end of June, I believe. Her name is Kirsten Porgal. She was previously a manager at the Species at Risk branch, which is sort of a sister branch to us, also in Land and Water Division within MECP. From what I understand and from what I've seen already, she brings a lot of experience in environmental policies and programs, and just can personally vouch for the fact that I've really enjoyed working with her over the summer and into the fall, and we really look forward to continuing to move our key files forward. But on top of that, we had a change in ministers. So as you may have heard, with the cabinet shuffle at the end of June, a new minister was appointed to the Ministry of Environment, David Piccini. So as you can imagine, with both of these changes, it's been a very busy time for us. We've been very diligently bringing our new decision makers up to speed on all the important work, of course, that's done locally across the province under the Clean Water Act to protect sources of drinking water, and also some of the key files that we've had sort of in the pipes for a little while, and I know there's a lot of interest in that people are eager to see out. So we're doing our best to move those forward. And that, of course, includes the amendments to the director technical rules that I know have been sort of on the agenda, and Katie has discussed over the past couple of years, because we've been working on them for quite some time. And these have not been posted yet. We continue to make great progress, and we hope to hear a decision about that very soon. In the meantime, we're also working on some guidance. I know there have been a lot of discussions with project managers over that, over the course of the two or so years that they've been developed and consulted on. So I would hope that, Katie and other project managers across the province have had a chance to be engaged and to ask questions, but we know there's still going to be probably some outstanding things that need to be worked through and to support, our those who are going to be implementing the rules when they are posted. So we're working on some good guidance. And another key project that's not finalized yet, but it also relates to what Tom was referring to earlier about protecting other types of systems, so non-municipal residential drinking water systems. So from what you recall, I think it provided more comprehensive updates in the past. We refer to them as sort of the best practices for source water protection. It's a document that we would hope would be, or not hope, anticipate will be posted on ontario.ca, and it's really sort of the easier, more cost-effective, straightforward strategies that local communities, First Nations, community leaders can basically put into effect in order to protect other types of systems that haven't been brought under the source protection plan formally. So those, we're working on finalizing those, and again, just very close to, I think, having a final product that we can post, and just working through some final details, but really, excited to hopefully see some movement on that soon. And that's it for me. If there's anything else that, Katie, that's top of mind that I might have missed, but.
Okay,
No, I don't think so. I think we're good.
Thanks very much, Olga. Any of the committee members have any questions they want to ask? Olga, who's representing our division. Okay. So we're at the presentations. Katie, I'll have you take it over now.
Sure thing. So I want to also acknowledge we have a couple of guests. So we have Emily Awad is here from the Ministry of Environment and Environment Conservation and Parks as well, and she's our local ministry representative here and works on the harmful algal bloom stuff. So Emily is here to listen to the presentations, and if we have any questions about what her role is, then she can sort of make note of those and provide us with a response. And we also have Deb Balika, who's joining us from Conservation Ontario. And Deb is the source protection lead at Conservation Ontario. And she's relatively new to the role and just taking the opportunity to hear and see how different source protection committees operate. So welcome to both of you as well. And Heather, of course, is here with us as well. So I'm going to hand it over to Chelsea Salter. So Chelsea is a master's student at the University of Windsor. And Chelsea's undergraduate and graduate work is looking at the Pelee Island sand, and as Kyle and I have referred to it as the magic sand on Pelee Island, where the water from Lake Erie just makes its way through the sand filter, and the microcystin that's in the raw water is no longer in it. So Chelsea's job was to figure out why that is. So Chelsea, I will hand over to you if you want to start sharing your screen.
Okay, thank you, Katie. Let me just share my screen really quickly. Okay, are you all able to see my screen now?
Yep, looks good. Thank you.
Okay. Yeah, so good afternoon, everyone. Thank you for the invitation to present today. It's an honor to be here. But like Katie said, my name is Chelsea Salter. I'm a master's student at Great Lakes Institute for Environmental Research at the University of Windsor in Dr. Chris Weisner's lab. And I'll be presenting my work that is titled Investigating Microbial Microsystem Degradation in Lake Erie Beach Sediment, which, like Katie said, was a project that I started in my undergraduate as a fourth year thesis project and has now been extended into my master's thesis, which I'm doing now. So just to begin with some background information, harmful cyanobacteria blooms or cyanohabs have become a serious global environmental problem in water bodies across the world, largely due to the effects of climate change and unsustainable nutrient loading. And there are several negative problems that arise from these blooms that threaten water quality and utility. However, one of the greatest concerns is the release of microcystin toxins. And so microcystins are a family of potent hepatotoxins that are produced by cyanobacteria and then released into water bodies, which exert severe animal and human health hazards due to their ability to inhibit protein phosphatases. And so over 200 different microcystin variants have been identified. However, the most common and most toxic analog is MCLR, where the amino acids leucine and arginine occupy the variable positions, which are colored on the slide that you can see here. And so the trouble with microcystin is that the cyclic structure enables it to be resistant to many natural factors that normally break down compounds. And while current water treatment methods do exist, these techniques are often unreliable, expensive to operate, and have the risk of potentially producing toxic byproducts. And so as a result, there's a great need for a technique that will eliminate microcystins in a safe, efficient, and cost-effective manner. And the leading solution to this problem is through microbial degradation. And so many bacterial species capable of degrading microcystins have been discovered, but they can all be separated into two groups, those that have an MLR gene operon and those that do not. And this is because only one microbial degradation pathway has actually been elucidated to date for MCLR. And so the enzyme encoded by this MLR gene operon sequentially hydrolyzes MCLR into less toxic byproducts through the mechanism that's shown in this figure. And it's generally accepted that the MLR genes that correspond in the corresponding degradation mechanism are not ubiquitous in all aquatic environments around the world. And it has been suggested by several studies that an alternative degradation pathway is likely to exist, but is currently unknown. However, despite many advances in the current understanding of microbial degradation of microcystins, it's still very rare for new degradation pathways to actually be proposed. And the ones that are propositioned are not fully supported because they don't have enough data to support their theoretical pathways. And so from these insights, it becomes clear that multiple genomic tools are required to understand the full complexity of the breakdown mechanism and to piece together the puzzle of how this degradation is actually occurring. And so my work specifically hones in on Lake Erie, which is especially susceptible to harmful algal blooms and has consistently sustained severe bloom events over the last couple of decades. And like many other lakes, microsystem biodegradation has been observed and documented in Lake Erie, but it has been argued again by several studies that the degradation is not the result of this known MLR gene pathway that I just mentioned. And so during a field experiment, I believe it was conducted either 2017 and 2018 on the shores of Pelee Island, it was observed that the potential microcystin toxins that were present in the lake were no longer detected in the groundwater after it had been filtered through these sand trenches on the shores of the island. And so the absence of the microcystin in the groundwater during bloom events suggested either a physical or biological elimination strategy was being employed by the sand at this location. And this actually contrasted with a lot of other studies at the time, which isolated the microcystin degrading bacteria from the water that was near these cyanohab blooms themselves. And so I was able to investigate this phenomenon in my undergraduate thesis project through designing a column experiment in which a stepwise increasing dosage of MCLR was spiked into flow-through sand columns. And so over the course of 6 weeks, water was continuously pumped through 6 sand columns, 3 of which contained sand that was unmanipulated from Pelee Island. And we referred to these as our biotic columns. And then 3 columns that had sand that had been gamma irradiated to act as a sterile control. And we just referred to this simply as our abiotic columns. And so water samples were collected several times a day during this experiment. And then sand samples were also collected weekly for RNA extractions. And so from this project, we were able to determine that the microbial community within the sand collected from Pelee Island was in fact very capable of degrading MCLR and could do so very effectively. And so in this graph here, we can see that all of the MCLR was removed from the system within 21 hours of contact time. And then another notable observation was that the abiotic columns actually developed the capacity to degrade MCLR over the course of the experiment, which we attributed to just an incomplete sterilization at the beginning of the experiment. However, this did end up providing an interesting aspect to the experiment because the abiotic column communities developed a separate and distinct microbial structure that was able to be compared to the natural composition in the biotic columns. And so 16S rRNA sequencing of the active microbial community showed that the most dominant taxa at the family level was Burkholderacea in both the abiotic and biotic samples. However, in the biotic samples, or as you can see, the biotic samples had a much greater diversity and was in fact able to degrade microcystin with a greater effectiveness than the abiotic columns, which suggested that several critical species may actually be required for the most complete degradation of MCLR to occur. So using differential abundance tests and supplemental literature review, we were able to identify a couple other family taxa that provide potential relevance in the degradation pathway of MCLR, which includes families such as Alumitobacteriaceae, Pseudomonaceae, Rhodocyclaceae, and Nitrosomonaceae. And then so now moving on to my current work, like I mentioned earlier, the elucidation of the degradation mechanism is a puzzle and it requires many different aspects to be pieced together to allow this full picture to come into view. And so building from the knowledge that was gained in my undergraduate program, my current project now aims to use a multi-genomic tooled approach to gain a deeper understanding of the mechanism occurring during this degradation of MCLR and answer some of those questions that were kind of left unanswered in my former work. And so with that being said, the objectives for my current work are to determine the capability of the Pelee Island sand microbial community to degrade parvalent microcystin congeners outside of MCLR, both separately and together, to establish the short-term kinetics of that degradation reaction through frequent sample collection over 24 hours of inoculation, to define which bonds are being targeted during degradation by analyzing the metabolites that are produced, and to finally identify the microbial taxa and diagnostic gene clusters that are actively involved in MCLR degradation. And so as opposed to doing a column experiment, this time the experiments were being conducted through simple batch experiments in which the Pelee Island sand was placed into 60 milliliter jars with filtered lake water. And then at the start of each experiment, MCLR is added to the water, and then the water samples are sampled over 24 hours which are then analyzed through liquid chromatography with tandem mass spectrometry. And this is to analyze the microcystin LR concentrations at each sampling point. And then we also ran 2 controls during these experiments, the first being a serial control, which uses gamma-irradiated sand to determine if decreases in the concentration could be attributed to physical absorption as opposed to biological activity. And then the second control is just a jar with no sand in it. And this was used to determine if any microbes in the water could be also contributing to this degradation. And so our preliminary results show that the Peely Island sand is able to degrade MCLR with high efficiency. And we were able to determine the rate of degradation reaction with a greater resolution than before, since the mass of the sand in the jar was only a fraction of what we used in the column experiments. Additionally, as you can see, the controls remain pretty stagnant throughout the experiment. So this kind of supports that these decreases in concentration are solely attributed to microbial degradation. And then here's just another plot showing the percentage of the original microcystin concentration that had been degraded at each sampling point. And it's important to note here that the initial concentration that were added to these jars was 10 parts per billion, which is actually much greater than natural concentrations that we see during bloom events. But despite these unnaturally high conditions, the microbial community was able to degrade over 90% of the original concentration within 24 hours. And so for the past year, I've actually just been repeating and revising my experimental procedure in order to optimize the conditions to obtain quantifiable RNA yields, which have been very difficult given the sandy medium that I've been dealing with. And now that I've been able to obtain sufficient RNA yields, my next steps will be to collect and extract RNA from the Peely Island sand to be sequenced for metataxonomics and metatranscriptomics. And then this information will be paired with metabolomics to provide a comprehensive picture of which microbes are degrading the toxin, which genes are being expressed in the process of that, and how the toxin is actually being broken down into smaller, less toxic byproducts. And then I also plan to expose the community to several other types of microcystin that are prevalent in Lake Erie, such as MCLA and MCRR, to determine the microbial community's ability to degrade congeners other than microcystin, which will provide some useful information in future applications of this study. And so just to close, with the incidence of cyanohabs predicted to increase, development of strategies and treatment methods that mitigate the most harmful effects becomes imperative. And so the results from the study will work to significantly improve the current understanding of microbial degradation of microcystins within Lake Erie, and will serve to demonstrate the potential utility of sand as the most suitable medium in biodegradation removal techniques, which can then go on to potentially be implemented in water treatment facilities and to streamline the removal of microcystin toxins from lakes and other bodies of water that are commonly affected by toxic cyanobacterial blooms. And with that, I'd like to thank you for your attention, and I'll take any questions if there are any.
Awesome. Thank you, Chelsea. If I can, I'll try to summarize a little bit, because there's a lot of, first of all, congratulations on learning how to say all of those microbial names.
I don't think I say them correctly, but
well, you made it sound very good and very convincing. So basically the ultimate finding for you is, so you had the sand from Peely Island just as it was, so with the microbial community, so that would be any bacteria, any little living organisms in the sand, compared against sand that had all of those living biological agents irradiated and killed. So it was just the sand. So the first finding is that we know it is a biological contribution to the sand and not the physical aspect of the sand itself. Mm-hmm. And then the next steps then are to figure out which genes they're using and which species exactly are responsible. Is that mm-hmm. sort of fair?
Yeah, because like what I did in my undergraduate was just kind of looking at the taxa, which you can see, like it gives a nice chart of like, okay, like these species are expressing RNA during this degradation process, but it's hard to like, it was difficult when analyzing it to be like definitively, okay, like is this actually what is contributing. So that's why like now it's like we need more information, more aspects that can support the data and kind of work together to, like I said, like allow that more comprehensive understanding of how it's actually being degraded.
For sure. And do we know if the community is unique to this sand? Or is that not yet? We don't know that yet.
I don't know that yet, but I think like when I was looking up, potentially relevant species that could be contributing, there was ones that we pointed out that have been or that were reported to have been degrading microsystem before. So that obviously was like something very notable for us. But I think the community as a whole, we don't know. Yeah, we haven't looked into whether that's unique in itself.
Does anyone else have any questions for Chelsea?
I do. Do you think that community was established before we started having huge problems, or do you think it was a result of those problems? Like kind of which came first, the chicken or the egg kind of thing? Just curious what your thoughts are.
Yeah, obviously like it's hard to like know that definitively, but I guess my prediction would be that it was a result, because it isn't like if you look at the island itself, it's right in that western basin, and the shoreline that we took the sand from is constantly being exposed to those bloom events. So we just figured over time, because of that increase or that constant exposure year after year, it probably adapted and found a way to like utilize that to its own metabolic benefit. So that's my prediction.
Mm-hmm. Yeah, and certain environmental factors can cause an organism to express genes that it may not otherwise express. So that could be part of it as well. Ron?
Were the other beaches or the other on Peely Island tested in the same manner as this one beach?
No, but that's an interesting question, because yeah, like it would be interesting to know if this is like a uniform thing, or if it's like that specific spot on that west coast of that shore that does have that ability. Yeah, it's a good question, but no, I don't know for sure.
That's a perfect thing. Every thesis needs to end with, here's what you should do next. So
Yeah.
it's a perfect, here's what to do next.
I'll keep it in mind for when I'm writing mine.
Awesome.
Chelsea, thank you for the presentation. One of the questions I had was, when doing your column study, the experiment using the columns of sand and water, did you notice any kind of decrease or possible increase in the ability of the community to remove and do its job, or is that something that you've been able to look at? And I guess the second question, kind of adding on to Chris's question, is would you see, or would you be able to see the community itself grow as it goes through your tests and as it's doing what it needs to do to take apart the bloom?
Yeah, so with, I guess, my column experiment, it was difficult to see, like, especially with the biotic columns, it was every concentration that we gave it, essentially, whether it be very low or extremely high, it would just like degrade it like within our first collection point, which was at 21 hours. So yeah, it was difficult to kind of determine how that community was, if it was growing or if it was shifting or whatnot. So like that's why these batch experiments are kind of designed more to sample within that 24 hours more, to first get the degradation kinetics of it and the reaction rate. But then when I do my RNA extractions to look at how that community is shifting as well on a shorter term. Yeah, I think that like my own experience, I guess I would say that like, because we are exposing it to such a high concentration, that like the community is going to adapt and shift so that it is optimized to degrade, like it's going to use that carbon source as its main like metabolic source is kind of like my prediction for what's going to happen. Hopefully that answered your question. If I missed something, just let me know.
Oh, Aaron.
Yeah, sorry to throw a last second question at you. I mean, you've done a great job answering so far, though. But mine, I was just curious to know if we're talking about a community standpoint, looking when you mention RNA, have you ever thought, so are you familiar by any chance reverse transcription PCR to identify gene expression? And the reason I guess my mind is going to there is that as you see this shift in the community, the behavior, so from a resistance or an ability to metabolize something standpoint, do you think you would be able to use the gene expression of the proteins for expression on that type of activity to be obvious and line up with what you're finding in your biotic columns?
Yeah, so are you talking about qPCR like specifically then?
Yeah.
Yeah, so I would like to kind of like implement that, at least in part, because I know like even just to rule out, like there are, like I said, that those MLR genes that are known to degrade the toxin. So even just to have like those genes and use qPCR on this community to see like if it's active. But I think at this point, that's why we're taking like a meta-transcriptomic approach. Like we don't know exactly what we are going to see. We have some like predictions, but we do want to see everything that's going on. And then, you know, hopefully we can get to a point where it's like we can identify specific genes that are being activated, and that can even be used as like a tool to, you know, identify, I guess, when degradation is occurring by specific species, or yeah. So I guess just to answer your question, yeah, we don't have specific genes at this point, but we would like to get to that point.
And Chelsea's got like the best of the best at Windsor help on her team to help out with the meta-transcriptomics and all that sort of stuff. So it's definitely on everybody's minds is, you know, is there a gene that's being expressed specifically by these microbes that is, you know, that isn't being expressed elsewhere. So
yeah, and I apologize if I threw you off at first when I said reverse transcription. I think RT, I should have said real-time PCR because you're talking qPCR. So
you guys can nerd out later. Well, you guys asked really amazing questions. Thank you for that. And Chelsea, I hope you found that helpful as well to see how important your work is and that the community is interested in learning about it. So thank you for sharing that.
Yeah, thank you again for inviting me. It was great.
Awesome. I'm going to turn it over to Emily Varga now. Emily is a PhD student at the University of Windsor at Clear as well. And she's looking at investigating the factors that drive the harmful algal blooms in the Thames River and in Lake St. Clair. So kind of starting to move away from Lake Erie. So Emily, if you're ready, I'll hand it over to you.
Thank you, Katie. Let me just share. And of course, somebody just came to the door, but everything should be fine. Okay, can you see that?
Yep, you're good.
Okay. Hi, everybody. Good afternoon. Thanks for being here. Katie, thank you for that great introduction. And to echo what Chelsea said, I'm also honored to be here. Excited to talk about this. It's very preliminary, so first chance I've actually gotten to discuss it with a group of people. I'm working with Dr. Mike McKay at Clear, University of Windsor. And as Katie said, investigating factors driving HABs in the fluvial lacustrine continuum. Okay, so give a little problem background why we chose this as our project. We're focusing on the harmful cyanide blooms as they posed a serious threat to our freshwater resources for decades. These blooms are dominated by hepatotoxin producing Microcystis in Western Lake Erie and in Lake St. Clair, and there are also various filamentous species of algae. Toxic strains of cyanobacteria pose a danger to people, pets, and wildlife. They can also alter the food web and create hypoxia. And for our project right now, we're specifically focusing on the fact that the community composition will shift with nutrient and light limitation. So to give a little bit of background on what's been studied so far and where there might be gaps in the research, it's common knowledge that this threat to our freshwater occurs globally by these toxin-producing blooms, and they're a regular occurrence in Western Lake Erie. But only more recently, we're seeing reports of bloom events in the tributaries. There have been many studies of Microcystis blooms in the offshore Lake Erie regarding environmental drivers of growth and toxicity. But there are other algal strains that are not well studied, such as Planktothrix. Also, tools of systems biology have only been recently employed for more in-depth study of these blooms, and these combine environmental chemistry, ecology, and molecular biology. So because it's been confirmed that the taxonomic composition of the blooms in these rivers are different from their downstream receiving waters in the lakes, we want to ask ourselves, do the rivers directly seed these lake blooms in terms of taxonomy, or are they contributing to the blooms by delivering nutrients to the lakes? Similar studies of Lake St. Clair have demonstrated that there are differences in primary production, chlorophyll A, and taxonomy relative to the difference in nutrient levels that it receives from the Thames River. There are also recent studies that look at the differences in structure and function of algal strains as a response to variations in nutrient enrichment in whole stream ecosystems. So by whole stream ecosystems, we mean looking at that river to lake continuum. We want to highlight the importance of understanding the factors that cause the differences in the taxonomic composition between the riverine systems and the lakes that they deliver nutrients to. For example, the findings of a 2019-2020 study identified filamentous algal species in the Thames River bloom. Specifically, Phanazomenon was the dominant taxa, and there's a picture of that on the right. Planktothrix agardii was also abundant in the bloom, sitting there on the left. And the conditions which promoted the growth of this bloom were low river discharge and very little rainfall. However, the blooms which occur in the receiving waters of Lake St. Clair are usually found to be dominated by colonial Microcystis. This one proliferates in eutrophic and hypereutrophic conditions, and this can be caused by agricultural runoff coming from the river. However, all of these different species have the capability of producing toxins, not just the Microcystis. So this leads to the objectives of our study. We want to examine which environmental factors promote or constrain the proliferation of algal blooms, both the toxin-producing kind and the non-toxic producing kind. For example, we want to look at what nutrient concentrations, temperature, pH, and light penetration cause these differences. And we want to examine the dominant algal taxa found in the riverine systems and see how they compare or differ to the downstream lake waters. We want to know the prevalence of toxin production by the blooms in these systems and if they vary by season. And because it has been shown that the cyanobacterial community will shift with nutrient and light limitation, we're going to generate our hypothesis around the idea that the dominant taxa within our study sites will vary with respect to changes in the environmental factors. For example, as shown in the picture, Planktothrix dominates in deeper, darker waters mixed throughout the water column, whereas Microcystis will thrive in the warmer upper waters of Lake Erie, where it can form a mat and reach all that sunlight. So we've hypothesized that the taxonomic composition of these blooms varies over time, both as a seasonal comparison, as well as between the rivers and their downstream lakes. So I have to note this project is in really early stages. We kind of had a wrench thrown in our plans with approvals through the university because of COVID. So there isn't much we've been able to do so far. So we're focusing right now on strictly the effect of light penetration. Which brings me to my methods. So the methods we'll be employing to meet these goals include water chemistry analysis, microscopy to identify which taxa are prevalent, and transcriptome analysis to assess the cyanobacterial response to the environmental factors. We will also eventually be getting to quantitative PCR and real-time quantitative PCR to determine the prevalence of toxin-producing taxa. So what we've been doing is we've been collecting weekly samples from the Stony Point water treatment plant intake well, recently also from Bell River. But Stony Point specifically, that treatment plant is right on the southern shore where the Thames River delivers a plume of nutrients to the lake. So starting this past June, we deployed in situ mesocosms, which is what you're seeing in these pictures, in the Thames River, where the epicenter of the river bloom has occurred in the past few years. So to begin, we used a handheld YSI sonde to obtain general water quality measurements of the river itself. And then we also pumped 100 liters of river water into a, sorry, there are construction workers here. We pumped 100 liters of river water into a clean garbage pail, which is what you're seeing in the second picture. And that was so that when we filled each mesocosm with water, it came from 1 ×0 sample to eliminate any other environmental variables. So the jugs themselves, we then manipulated in regards to how much sunlight they were exposed to. And I did this by sewing fiberglass window screening around them. So we had a control set of, everything was run in triplicates where there was no window screening on it. And then 3 that had a single layer, 3 that had a double, and 3 that had a triple, so that we were slowly increasing the light attenuation. And we put these in a little enclosure that we lovingly named number 7. And we floated that behind the Lower Thames Valley Conservation Authority, attached with a steel tether, so it's not going to float downstream and pollute with all the plastic. And this way it was exposed to the same amount of sunlight and temperature as the river water in general. So the first day we brought back samples from the ×0 from the garbage pail, did all the analysis, well, starting to do the analysis and the processing for all that, all the chemistry, all the nutrients. We've sent samples off for the microscopy. And then we returned every 2 or 3 days, depending on the weather, collected a liter out of each one, and then did the same sort of processing. And then we pulled it out 9 days later, and then repeated the entire process in late July and mid-September. We are also filtering some of these samples and freezing them to quantify the toxins and pigments, specifically microcystins and phycocyanin. And not to be too repetitive after Chelsea's presentation, but we know with microcystins, there's more than 200 congeners, and microcystin LR is the most toxic. The reason it's so toxic is it inhibits protein phosphatase activity in the liver. And the reason we look at the phycocyanin is it's a way to estimate cyanobacterial biomass, as it's an accessory pigment to chlorophyll. We also deployed a little pilot project this year in the Stony Point water treatment plant intake well. This was an experiment to see if real-time monitoring would work. So we dropped this sonde in the intake well, which you see in the bottom picture, connected it directly to a data logger, which recorded all the measurements every 10 minutes. So we looked at pH, specific conductivity, temperature, chlorophyll A, and blue-green algae. And then the data logger communicates with the LoRaWAN gateway, which you see in the top picture, which is basically a cellular modem that projects that information to Limnatech over in Ohio. And then we have a real-time dashboard where we can see all those measurements every 10 minutes. Unfortunately, there are a lot of problems with this type of equipment, like Ethernet cords don't like moisture, and we had it in an intake well. So there were a few times we had to go out and change things and fix things. And it actually just quit on me a few days ago. So I was going to show you the real-time dashboard, but the site itself has been acting up and it's not up to date. But if there's time at the end, I can go back in and try to go online and look at it. If I try to do it through my presentation, I might drop the presentation. So, okay, and I do have some results to show, which is exciting. I haven't really analyzed this yet. I haven't run any of the stats. I literally put this graph together yesterday because we just started getting it all back, but to show you something. So at the phytoplankton biomass, this is time 0 and the first time we went out at day 2. Interesting to note that at time 0, there was a lot of picoplankton, diatoms, chlorophyta, and there was some cyanobacteria. But then looking at the control and all 3 treatments 2 days later, it was mostly diatoms. Again, that's just very preliminary information. Looking at the nutrients, looked at the mean soluble reactive phosphorus, or the most bioavailable, and the total phosphorus at time 0 and day 2. And then also compared to time 0 to day 9 when we pulled the whole thing out. And I think the only thing to note here, without looking at the stats, is that there is a depletion in the SRP and somewhat in the TP over that time period. Then the nitrates and the ammonia, again, just to show the time 0 and day 2, and then time 0 compared to day 9. There's a lot less depletion of the nitrates. Again, I haven't run stats, so don't quote me on that, but just from looking at the graph at this moment, it's not telling me a whole lot, except that my diatoms don't like nitrate. Also looked at the chlorophyll A. No distinct pattern here yet, but interesting to note that at the single screen mesocosm, the 60% light attenuation, the chlorophyll jumped up. And I think that might have just been the algae cells trying to adapt to their new environment. I guess the less sunlight you're getting, the more chlorophyll you're going to put out there to, try to catch what you can. So for the implications of the study, we're hoping to develop the tools to predict the composition and quantity of future blooms in terms of the taxonomic composition and the potential of those species to produce toxins. In other words, hopefully this will help with bloom mitigation. It's beneficial at many levels, including municipal, provincial, and public health concerns. For example, the Ontario Health Unit being responsible for monitoring the beaches and notifying people when it's not safe to swim, as well as the Ontario Ministry of the Environment, who regulate our drinking water. I would like to acknowledge my committee members, my advisor, Dr. Mike McKay, Dr. Katie Stamler, Dr. Catherine Fabria, and Dr. George Buller-John. And I also want to shout out to my sampling team. Again, because of COVID, I was not allowed to have lab mates out in the field. So my family stuck with me the whole summer and helped me through this. I also want to mention Great Lakes Center for Freshwaters and Human Health at Bowling Green State University, where all of my chlorophyll samples were run, and the folks at Limnotech who set up the dashboard for me. And that's all I have for you. Thank you.
Awesome. Thanks, Emily. I was looking at your pictures and was like, those field assistants look really young. We do what we have to do these days.
Child labor.
That's awesome. And so Emily's work is going to help us understand, and you're going to see the data that we're going to look at for Lake St. Clair is going to help us understand how those blooms are happening in Lake St. Clair and what the contribution is from the Thames River, and then help us start to understand what we do about it. And the real-time songs, that part is exciting as well. And there's some work happening with Rick Stumpf as well with NOAA to look at how do we take those predictions that he's doing for Lake Erie and also expand them into Lake St. Clair. So doing a little bit of checking of the data and making sure the satellite images line up with the data that we're actually getting. So that'll be an exciting product at some point. Does anyone have any questions for Emily? Ron's good. He's happy.
Thank you. I'll take a thumbs up.
That's great. So yeah, so like Emily said, her work is brand new. So we will probably hear from Emily again as she continues on with her research. So if there's no questions, we will let Emily go.
Thank you.
Thank you, Emily. Okay, so the last person that I invited to speak today is Kyle Davis, and he is currently the Water Compliance Coordinator for the municipality of Lakeshore, previously of Pelee Island. So Kyle is well versed in being a water treatment operator in a place that has harmful algal bloom. So makes sense for him to be in Lakeshore now. He's only 3 months to Lakeshore, but a veteran of harmful algal blooms. So, and Kyle and I go way back in dealing with Peely Island issues. So I'm going to hand it over to Kyle. Kyle's going to talk to us about how a water treatment operator operates during a harmful algal bloom.
Thank you, Katie, for the warm introduction. We have worked long and tirelessly on this issue with water treatment over the years, together, along with many other people, with the MECP, back then the MOE, the Windsor-Essex County Health Unit, yourself, your sampling teams, and all are all valuable assets to water treatment plant operators if utilized correctly. For everybody, my experience starts back on Peely Island. I was the environmental manager for the island, for the town, was involved in building a water treatment plant specifically for blue-green algae and the design aspects for it, with now retired Gary Johnson from the Ministry of the Environment. Emily Awad was involved earlier in her career. Mark Bayshard, yourself, Katie. A lot of individuals were tasked, along with some very good engineering, to build probably one of the few plants specifically designed for that problem, and being modular in design so that as problems started, they could be dealt with as different treatment phases had to be brought online, with cost being of concern for a small municipality. We noticed with the Peely magic sand, let's call it, the term was coined, and I can't remember the gentleman's name from Environment Canada, but having discussions with him back in 2012, 2013, after the bad 2011 year, of us sampling for microcystin toxin in the raw water with our DWISP samples, drinking water surveillance program samples from the Ministry of the Environment, and having concentrations of 20 up to 50, I think just over the 60s mark was our highest in the lake water. This is pre-treatment of any kind. And then through this infiltration gallery, the natural sand lying on the shoreline, and then sampling the raw water clear well, finding nothing. And that triggered those investigations. And it's just how important water treatment plant operators' observations can be, and have triggered Chelsea's studies today, possibly Emily's studies today, all from those questions being asked up the chain. Water treatment plant operator knows how to clean water, but if he doesn't know what he's trying to clean, then it makes it very difficult, or the toxicity basis of that. Being water soluble makes it very difficult. On Peely Island as well, it's the highest concentration of small drinking water systems in the province, dealing with the Windsor-Essex County Health Unit there, because they mostly utilize small shoreline infiltration wells, just like what Chelsea was sampling from in her studies, and determining that those well systems, because of the natural sand through much sampling, a lot of sampling, that there was microcystins determined in the lake and at various concentrations surrounding the island, and none was ever detected within their raw water clear wells. So those types of things are very important, and all that was drummed up from a simple question as to observing and understanding your samples as being a water treatment plant operator, and then knowing how to adjust your processes after you take it to someone that may know, whether it be the science-based community, whether it be the governmental agency-based community, someone will know the answers. And that's why the Safe Drinking Water Act particularly has that safety net in there for water treatment plant operators to ask the questions that if they don't know, they're not going to get in trouble for asking the question and have it come back down. Someone is generally going to know the answer on how to proceed with a certain treatment phase. Peely Island, it was sand infiltration to membrane, seaweed membrane filtration, and then should issues arise, cartridge carbon filters were put online, and then increased standing contact time with chlorine for oxidation purposes. So there's a multi-barrier approach within the Safe Drinking Water Act that all water treatment plant operators and all water treatment plants have to keep in mind when treating for everything. And microcystis and microcystin are just one of the phases of things that we treat on a daily basis. Good thing for us, it's just seasonal for the microcystis and microcystin. From that, the plant was built there, and I believe there hasn't had a problem since. One thing that was interesting there, and because the Ministry of the Environment helped out that small municipality so well in their sampling costs, we were able to sample and determine that actually a dirtier filter bed helped the removal of any microcystin should it enter the treatment plant. And that has been up for discussion amongst treatment operators for many times, whether the biodegradation filter seeding is a potential aspect for future treatment phases. And Chelsea's project and her studies are starting to prove that could be something to look at in the potential near future, but more likely down the road, that it could be a viable option. I can safely say that I've worked in every watershed within IRCA. The southwestern basin of Lake Erie is fed by the Maumee River system, and that's why Pelee Island is such a unique position and so overwhelmed with toxic microcystin blooms, is because of that different water than the Detroit River and the northern watershed of Lake Erie. I worked at the Union Water plant in Ruffin under Rodney Bouchard and Dale Dillon there, both wonderful mentors, and the way that they have that facility set up and prepped for microcystin removal as well. Totally different treatment phases, but they've been able to optimize them based on water treatment plant operators' observations on a 24-hour basis. Management is made aware of the problems should they be arisen or possible weak points, and they've adjusted for that, and they continue to make upgrades there for that. But once again, it all comes down to your water treatment plant operators being able to observe any abnormal observation and immediately report it to someone that can take care of the problem before it becomes a bigger problem, especially for the populations. Now working up here in Lakeshore with 2 water treatment plants that have 2 totally different treatment phases, but are taking water from the same source, Lake St. Clair. It's interesting to see how the same source water, just in 2 different locations within the lake, can be so different. And working with Emily, we're learning that, and hopefully going to learn more about that so that our 2 plants can optimize better in the future through our observations. One thing that the operators do here at the Bell River Water Treatment Plant where I'm stationed is when algae's identified, the sodium hypochlorite injection is taken offline to stop the degradation and lysing of the cells to potentially release any microcystin into the treatment system. From there, we have the potential to add activated carbon should it be deemed bad enough. The plant never has had to in the past, but it is a source and it is a treatment phase that is potentially, and it's tested monthly and able to be put online at a whim should an operator make an observation that is not normal, as something in the computer system or some alarm or just some observation of a weekly trend that he's been monitoring. We can change the treatment phase. We also increase or decrease our run times, increase our backwash times. So instead of a 200 or 400 filter run time, we may drop it down to from 400 to 200 down to 100 hours, even lower if required, if we notice that our filter beds are loading up. And this can be just a visual observation. As well in our clarifier process or sedimentation in our coagulation process, we can notice what amount of algae or diatoms are settling out and how our process or versus actually forcing through the coagulation process and swimming to the top. Floaties versus Sinkies, little simple thing in the water treatment industry. The floaties are more representative of a blue-green algae trying to get to the light, trying to get up to survive. And those are now what is going to make it, because they have swam through the coagulation bed of alum, are now going to be enter your filter process. So you can see in your daily or your twice daily samples collected of what sludge you are removing versus what is getting transferred over to your filters. UV system potential has used potential upgrades for advanced oxidation processes in a reservoir should we ever need a more of a process in our effluent side before it gets to customers. And that's the ultimate end goal. This treatment plant here right now is running at almost a 60-hour retention time in its reservoir. That is a very long time for that water to sit in contact with a higher chlorine dose before it goes to customers to ensure oxidation of a lot of these compounds. Some of them that we don't sample for regularly, but sample for on an annual basis that we know they're there, but it can degrade before it gets to consumer. can't stress it enough that water treatment plant operators are someone to listen to. Generally, they're very good at going up the chain of command to their supervisor, their lead hand, and then to management or to myself. In the compliance aspect, is this a compliance issue? We've noticed this. No, but thanks for letting me know. And we can go to management and see if we have to change anything. There's a lot of things treatment operators do on the fly, that they're the operators in charge of that plant for that day, and they can modify treatment processes to make the daily observations. It's a very scientific trade. We scientifically nerd out all the time in water speak, as Dr. Katie, and I'm seeing a few head nods here. But it's sad to get excited about those things, but we get pretty happy when we can find a problem and fix it. Because we're ultimately in charge of keeping our community safe, and a community is nothing without clean drinking water. So thank you very much for the time speaking, and feel free to ask me any questions about this. I'm probably one of the few drinking water operators in the province that have gone through a do not use water advisory issued by the Windsor-Essex County Health Unit in the 2014. After the Toledo crisis, Pelee Island had its own crisis, and our fire department had to go around there and to everybody door to door, and for a do not use water. You couldn't brush your teeth, you couldn't wash your dishes, you couldn't use anything. Unfortunately, there were some the pets that didn't do too well from the water, but it was a very scary time because the water was crystal clear. There was no present blue-green algae in the area, but the ball that was sitting off at Toledo had finally leached out into the water column and made the entire southwestern basin of Lake Erie toxic. And through the help of IRCA, Ministry of the Environment, Windsor-Essex County Health Unit, we successfully had no injuries from that incident and successfully got the message out. Crippled an economy for an entire municipality for a Labor Day weekend, but no one got injured. And that's the main thing. The main role of a water operator is safe drinking water. So thank you again.
Awesome. Thank you so much, Kyle. And I think, you'd be hard pressed to find another water treatment operator that's seen as much harmful algal bloom as Kyle has, and all these different ways of treating water. Kyle, a question that I have is like how during the harmful algal bloom season, it's on your mind, right? And you're watching it, and you're altering your processes and keeping an eye on processes during the harmful algal bloom season, correct?
Correct. We have a few key indicators. A few that I bring with me to every plant that I work at. One of them is pH. Mm-hmm. It's one of your first indicators. I learned that on Pelee Island, that the pH in the lake would go from 7.5 to 9.5 within a couple of days. And that's just a key indicator of the amount of carbon dioxide being removed from the water column, meaning there's a healthy algal bloom and they're contributing to the pH in the water. Your temperature is, once your temperature gets over 17 degrees, it's time to start thinking whether it be harmful algal or not, you're going to start having more biologic growth on your filter beds. You're going to be bringing in and possibly hampering your coagulation process. You have to change and modify your systems. And correct, it's the start of it all. And then your observations take place from there, depending on what you may be bringing into the facility.
Awesome. Anyone have any questions for Kyle? Awesome. Way to go, Kyle.
Thank you very much for the time. And anybody, please feel free to send me an email or get in contact with me here at Lakeshore. If you do have any questions for your municipality or algal blooms in general. So awesome. Thanks for the time.
Thank you, Kyle. So the presentations, all three of them, I wanted to bring to you. The only report that we have before us today is a report on microcystin in Lake St. Clair. So I wanted to invite these three speakers today to sort of set the stage for the scale of the issue of harmful algal blooms and microcystin in Windsor, Essex, and how lucky we are to have some research based locally on this work, which is really amazing because often the research happens outside of the area. So really great to see some interest from the University of Windsor in that. And also wanted you to hear from Kyle so that you hear about what has to happen from a water treatment operator's perspective during a harmful algal bloom. So thank you to all three of you for providing that context. Really appreciate it. So I'm going to walk us through the report. Oh, and Kyle, as he was talking about the upgrades, so the picture that I always use for our opening slide is the Peely Island water treatment plant, and shows some of the upgrades, the increased storage in their standing pipe there, so that they have lots of water available if they're ever in that situation again. So I'm just going to skip right ahead to our SPC report. So you had that report in your agenda given to you, and it's a bit of a hefty one. So lots of technical stuff in it, and I'm going to walk us through a couple of different things to do with it so that you have an understanding of what's in that report. So the first thing I want to touch on is that this work is happening. I'm proposing that it happens through our Section 36 update. And just a reminder that we have a couple of different options for making updates to our source protection plan. That Section 36 update is a mandatory review, which we did that review back in 2018 and made a bunch of, we proposed a bunch of updates, and this was one of them. So the Section 36 update is where we have that opportunity to change our existing policies, extend our vulnerable areas, add some new policies. So that's where we're going to propose that this work goes into. And I'll remind you again, at the end, there's a consultation process and all sorts of things. So we're not making a final decision here, but it's the beginning of this conversation. As I said, the Section 36 work plan we did back in 2018. And we created that together. We went through a lot of different reports that outlined the needs that the SPC addressed, and that work plan was submitted in 2018, and then we received a minister's order about the work to be done. So where the microcystin as a drinking water issue in Lake St. Clair falls is under proposed update number 2, where we outlined that microcystin was already identified as a drinking water issue for our Lake Erie intakes. We did that back in 2014. And we identified that we should make an assessment and complete an assessment to determine whether microcystin should be identified as a drinking water issue for our additional water treatment plants in Lake St. Clair and the Detroit River. We did at that time an interview, like a written interview with our water treatment operators. And at all 7 of our water treatment plants, our water treatment operators identified microcystin as something they consider to be an issue that they're keeping their eyes on and that they're watching for in the summer. So that water treatment operator input was very important in this. So we have this this approved, hold on, Bill Dukes show up here. So we have this issues evaluation methodology that was adopted, I think it was 2009. And I believe it's used by multiple SPCs, but this is the method that the Essex Region SPC uses to identify an issue. So we look at different sets of data. We look at those water treatment operator interviews. We look at the data from the drinking water surveillance program. And then we ask a few different questions. We have the first level where we look at, did the operator flag a concern? Do we have conditions above that maximum allowable concentration? Is it happening more than once? And if the answer to those things is yes, we move to the next stage and we look at whether the concentrations are above that maximum allowable concentration, whether it's trending towards it, how frequently it happens, how long it happens for. And again, that water treatment operator opinion. And if we satisfy enough of those, then we say, yes, this is a water quality issue. So that's the method that we're going to, and I'm going to work you through it with the data that I have to show you. So the first step is looking at the data, and we're going to look at that first thing. So flagging the operator concerns, we know that we have that, and then we're going to look at the data. So this first graph is from Bell River. So this is the average monthly total microcystin in the water. The reason we look at total microcystin instead of the specific congener microcystin LR is that it's a very expensive laboratory analysis to break it down into the congeners. So we look at the total, and we know that microcystin LR is the dominant form of the toxin. So we can feel pretty confident if we have high levels of total microcystin that we're also going to have high concentrations of microcystin LR. So this is the total microcystin in the raw water in Lake St. Clair, averaged for each month over several years, over about 10 years. And you can see that seasonal nature of the concentration. So it starts coming up in July through July, August, September, sometimes into October, and then goes back down again. The first dashed line at 1.5, that's the maximum allowable concentration. So that's the number that we're saying, oh, is it near that? Is it reaching that? Or is it, and then that half maximum allowable concentration. And you can see for Bell River that the averages are well over that. So we can feel pretty confident that we're flagging that, yes, this occurs above the maximum allowable concentration. For Stony Point, the concentration of microcystin is lower than it is at Bell River, but it is still frequently above the half maximum allowable concentration, which still flags for us that it is a drinking water issue in these places. So when we look at that first step of the issue evaluation methodology, yes, we have operator concerns. Yes, it happens above or at half MAC. And it's happening more than once because these are our average concentrations that are up above these numbers. So, so we say, yes, it's been flagged, and we go on to step 2. So we're going to look at another avenue of the data here. So this is the average total microcystin for the whole year over time. So we're looking at that trend and saying, you know, is it trending towards maximum allowable concentration? And there's no trend here in the data, but what is important is that it's always above the half MAC or close to the MAC. So it's not decreasing and it's not increasing, but it's also not changing. So, so we don't need to necessarily worry about it trending towards maximum allowable concentration because it's already there and it's not decreasing. So that's for Bell River. And then Stony Point, again, the concentrations are lower here, but still in that range that we can consider it above that, at least above the half MAC. And then the other question it asks is how frequent is the issue in the raw water, and how long does it last for? And we saw from that seasonal look that it happens throughout the summer months. These are a count of the occurrences. The top graph is above the maximum allowable concentration. So how many times was it above 1.5 micrograms per liter? And then the bottom graph is how many times was the concentration above the half maximum allowable concentration. And this is again for Bell River. And you can see that every year there's a number of occurrences above both of these levels. Again, for Stony Point, the numbers are lower. The interesting thing here that I thought was that you don't have that same in Bell River. You have a lot more data that's between the half MAC and the MAC, where at Stony Point, if it's high, it's really high. And it's always above the maximum allowable concentration. So again, we go back to that issue methodology and say, yes, we know it's at or above. It's not trending towards it. It's already there. So that's kind of, you know, neither here nor there. We know that it happens every year and it happens often throughout the year. We know it happens through the course of the summer. Kyle has just given you a really great conversation about the treatment plant capability. So we know our treatment plants are capable of dealing with it, but it is something they have to be aware of, which is the ultimate definition of a drinking water issue. And then the operator opinion. So then we say, okay, we've met all of those things. Yes, this is a drinking water issue. What I should have started with that I just realized is that I should have backed up and reminded you what the difference is between a drinking water issue and a threat. So a drinking water issue is when we have an actual condition in the source water that we know to be a problem for our drinking water. So in this case, it would be microcystin being the drinking water issue, versus a threat, which is an activity on the land which could create a contamination of the drinking water. So those are our lines of evidence. So what you as the SPC have 3 options that the ministry outlined for us back in 2014 when we were considering microcystin in drinking water for Lake Erie. The first option is to identify it under the technical rules, and there's some jargon in here, but basically if we do that, then we must identify an issue contributing area and we must identify significant drinking water threats. The second option, and I'm going to go through each of these and why they work or don't. The second option is to identify the issue under the Clean Water Act only. And that means that then we don't have to identify the issue contributing area. We don't have to identify significant drinking water threats, but we can write policies. The third option is to say that there is no issue. So I'm going to talk about the third option first. I've just shown you several lines of evidence that would make it challenging for us to say that there is no issue. So let's take that one off the table. Look at the first option. The problem with doing it this way, with identifying an issue contributing area, normally the way that the Clean Water Act and its technical rules were written is that most issues, you know what's causing that issue and where it's coming from. In this case, is what Emily's work is relevant to is it's massive. This problem is huge. It's coming from the Thames River. It's coming from multiple other places. It's coming from, watersheds throughout the Essex region. So it becomes very challenging to identify that issue contributing area. And we don't have that knowledge yet. I mean, that's what Emily is working on. So we can't really do the first one. So what we opted for with identifying the issue for Lake Erie was taking this second approach and saying that it is an issue. We're going to identify it as an issue under the Clean Water Act, but we're not going to identify an issue contributing area. We're not going to identify significant drinking water threats. And then that allows us to still have those policies, which we already have in place for those Lake Erie intakes. So that's what I'm saying to you as the SPC. This is my recommended option. And what we do once we make that decision is that I then will do some work in our assessment report, working on summarizing those data, updating, and then I will probably also update the Lake Erie data and the Detroit River data while I'm at it. I didn't look at the Detroit River data here, and I'm going to tell you about something that's going on in the Detroit River that's really cool. And then we're going to, look at those treatment concerns and incorporate all of that into the assessment report, update the appendix that we currently have. And important to note here is that there is still a consultation process to go through here. So the municipalities will still be consulted with, the public will still be consulted with. So there's still some various lines to go through, and obviously the ministry and their science coordinators. So that is what I had to share with you for the report. So these are the resolutions that I wrote into the report for your consideration. So the first one is that we identify microcystin LR as an issue under Rule 115.1, which is that option 2 that I described. And the second resolution is that the technical work or anything that's needed to be completed in the assessment report as part of Section 36 happens, and that we update our existing policies to include these Lake St. Clair intakes. So those are the resolutions before you, and I'll turn it over to Tom for that part.
Thank you. So we got to make the committee bigger so I can see it. There it is.
Yep, I did it.
So are there any questions or comments before we talk about the recommended motion here? Does anybody have any input? Tim?
Sorry, I just found my microphone button there, Tom. Just wondering, with respect to the technical work, are we going to have to ask for funding to get that work done, or what is the process involved to complete the technical part of that resolution?
It's not much. It's really what I just showed you with the graph. So that would be then working with George Jacob at the Ministry of Environment and making sure that we've covered all of the lines of evidence, which is basically just, making sure that we've got the appropriate graphs and data included. So there's no modeling work. There's no, nothing like that. So it's a small amount of my time. to do that. So that would be incorporated into our ask in our transfer payment agreement.
Thank you. That's all I had, Tom. Thanks.
Okay, anybody else? Some guys are off camera here, so I just want to make sure I give everybody a chance to chime in here if they want to. And I don't see anybody popping up.
I think Emily has her hand up. Emily Awad.
Oh, I didn't mean to put my hand up.
I think have we lost a couple of SPC members? One, two.
The part I don't like about this when the members that vote are identified somehow.
Yeah. John seems to be having some difficulty. He's
Yeah,
been
he disappeared. I know
he's
that.
been disappearing and reappearing. We've lost Aaron Corstein. So we are currently at
Oh,
9. Oh, and Kyle's dropped a couple times from the call. Am I at 9? Ron, Nelson, Kevin, Jim, Matthew, Cynthia, Chris, Tim.
are you saying that we can't have this vote?
Oh, dag nabbit.
Don't tell me that.
I've got 9. Sam, what are you showing?
Sorry, I'm just trying to scroll back and forth because they're separated between two panes of. Let me double check.
Oh, thanks, Emily. Yeah, John tried to come back, but he didn't make it.
Thought I counted 10.
No. Oh,
Jim, Bill, Chris, Cynthia, Kevin,
sorry,
Matthew,
technical glitch.
Nelson, Ron, Tim. Oh, 9. Okay. We didn't get any proxies, huh? Sam,
You're on mute, Katie.
you're muted, Katie.
Sorry, Katie, you're on mute.
Yes, I am. No, Antonetta did not provide a proxy unless she emailed somebody directly. I don't think she did. I did email John back. He just asked if we were done and I told him to come back to vote. Would it be totally unorthodox if I moved to the other business and came back in case John makes it back?
No, that's good.
Okay.
The other business being,
There is some fun other business. Oh, couple things. So Emily Awad has been sharing with us. There's been a couple of fuel spills recently. So they've been small, but they have happened. So it does remind us that the work is important that we're doing, and that that fuel is something that we do need to still consider. And the lines of communication are working. So when those fuel spills are reported, they are being reported then to the downstream water treatment operators who need to know about it. So just something I wanted to note, that that system is working. The other thing I had alluded to with Detroit River is the SEMCOG, the Southeast Michigan Council of Governments. They run a real-time monitoring network through the Huron-Erie corridor. So they've got all of this really amazing real-time data happening in, sorry, my pile of humans has just arrived home. So they've got this real-time monitoring network that's looking at phycocyanin, which is the pigment produced by microcystis. as well as lots of other things, DO and pH and temperature and all sorts of other cool stuff. And they've got a platform for that. So we just a couple weeks ago had the utilities from Windsor Essex participate in a meeting with SEMCOG to figure out whether we can join up with that network. So it sounds like the Windsor Essex utilities are very interested in doing that. So we're working with SEMCOG to figure out how we both access that real-time network and provide data to it. So really neat opportunity to work internationally with these shared waters. So that's something I thought was really neat that I wanted to share with you. The last thing I'm going to tell you about is that Conservation Ontario has a road sign communication campaign going on right now. And we made our very own roadside video. So you guys are getting the world premiere of the Essex Region Source Protection Committee roadside video. So I'm going to hit play and just start shouting if there's no sound to it, but it should work.
Have you seen these signs?
There are more than 60 of these distinctive blue and white signs across Windsor and Essex County, and they indicate that you've entered a vulnerable area near a drinking water intake.
Drinking water protection zones are the areas that provide the water that eventually becomes our drinking water.
We need to be extra cautious in these zones to ensure that our actions don't result in the contamination of our source water.
The Essex Region Source Protection Plan contains policies that provide extra protection for the sources of drinking water for our 7 municipal drinking water systems.
Which draw the water from Lake St. Clair, the Detroit River, and Lake Erie.
These signs create public awareness of the vulnerable areas around shared municipal sources of drinking water. They can be found throughout Ontario on roads and highways.
They also alert municipal responders that there's a source of drinking water nearby.
In the event of a spill, emergency responders can notify Ontario Spills Action Centre and local water operators. Quick action can protect our sources of drinking water and public health.
Your property, business, or farm is located in a drinking water protection zone. These signs remind us that our actions in these sensitive areas matter.
What we do here.
What we do here.
What we do here.
What we do here.
What we do here could affect the drinking water that we get from Lake St. Clair.
The Detroit River.
And Lake Erie.
Together, we can protect our community's water source. Protecting our sources of drinking water is the first step in a multi-barrier approach that ensures access to safe, clean, and sustainable water supplies from source to tap, now and into the future.
To learn more, go to our website in the link below. Keep your eyes open for drinking water protection zone signs all over Ontario.
Way to go, everybody!
Applause, applause, applause. Oh,
I'm super happy with how that turned out. So thank you to everybody
yes.
who participated.
All done by amateur photographers.
That's right. All self.
Well, mostly.
The actors look very experienced.
I've got John. Oh, John, are you in now? He's desperately trying.
How come my audio's gone way down? Is that you? Is that your fault?
Is it my fault? I don't think so. Can you hear me?
It's a bit hard to hear, Katie.
I don't know why. Can you hear me now?
Yeah, there you go. You said something on top of your microphone? Anyway.
I don't know. This microphone cuts in and out. So John is trying to join from multiple devices and doesn't seem to be having any luck.
Can he call in and be relayed somehow? I'd like to get this motion done if we can.
Yeah, no, for sure.
Can John give me his proxy?
He can.
While Katie's working on that, Chair Firth, if you don't mind, I just wanted to give a little update from an industry point of view.
Thanks, Matt.
Yeah, I know. I believe I had mentioned in a previous meeting some of the changes that are going on in the marine fueling industry. So in oil and gas, one of the biggest changes is the reduction of the allowable sulfur content in fuels and the requirement for transportation companies to reduce the amount of sulfur in their exhaust. There are two ways of doing that. One is to clean the exhaust using a scrubber, but the more common way is to use a cleaner fuel. Most companies are switching to marine diesel, but at Sterling Fuels, we have had one of our customers do a project all year using biodiesel, and from our point of view, it has been very successful. I mean, there are certainly a number of factors, but one of the greatest things coming from the point of view of a health, safety, and environment specialist at a fueling company is when I look at the effects of the products that we use and the MSDS, the SDS, looking at what we need to do to keep our workers safe. One of the things I will say when we're looking at biodiesel, the recommendation if someone were to ingest biodiesel is to consult a physician if you feel unwell, but otherwise there is no concern. It is essentially the easiest way to describe it is it's French fry oil, it's used oil. However, it does come from a number of sources. It can come from algae based, it can be plant based. Ultimately it all ends up being the same. But the fact is, it's one thing that I've had several meetings with regulators, the individuals that I deal with Environment Canada, and the first question that I asked is, who exactly do I need to call if there were to be a spill in the water? And the answer is no one. It's not a regulated product. There is still certainly the concern that it can cover the entire water area. It's something that could be an issue in engulfing organisms within the water. But as far as the danger to the surrounding environment, at this point, they haven't identified one. Now, that being said, knock on wood, we haven't had any spills. If we were to have a spill, I would still be contacting Spills Action Center and make sure that everyone knows, just because I'm not going to take any chances. It's nice to see that industry is moving in that direction. We are hoping for that next year, the bio business expands and more of our customers are using it. It's something that's good for the environment and certainly good for drinking water, especially when we have this amount of industry going through the Detroit River. So just a little update that I wanted to share with everyone, just to present that that insight from industry.
That's awesome. Thank you so much.
Thanks, Matt.
I have John on speakerphone.
Yeah, I got your note. Oh,
Perfect.
there's another note.
Nope, that's it.
Okay. Okay, good. Was John able to hear the motion and the stuff that led up to it?
John, did you catch the resolution? Okay, so the data presented was showing the levels of microcystin and meeting those requirements of the issue methodology, issue identification methodology, and the recommendation that we call microcystin a drinking water issue, as we did for the Lake Erie intakes. So the resolution before the SPC is that microcystin be identified as an issue under Rule 115.1, like we did for Lake Erie, that we complete any technical work that's required for to be included into the assessment report under our Section 36 update, and that we modify our existing policies to include the Lake St. Clair drinking water intake. So those are the resolutions before the SPC right now.
Okay, so committee members that are on screen before we lose John again. Somebody want to make that motion? Cynthia and Ron. Okay. Sorry, Jim. Cynthia and Ron. All right. Any question, addition, deletion, correction to the motion? All in favor?
And John, can you just? John has said he's in favor.
Okay, good. All right, great. Motion passes. Chris, did you have a question or you're still voting? Okay. I think that brings us to the end of the agenda. And I thank you very much for your patience with us getting quorum enough to get a vote in.
Yes, thank you.
After all that. So thank you for your patience. Oh, I didn't say hello to Larry. Do you have anything from the SPA you want to say, Larry?
No, not really. We're still holding our meetings the same way too. No, it's going good, and looking forward to welcoming our new member the next meeting.
Great, great. Thank you very much. Anyway, with that, is there a motion to adjourn? I think I saw Tim. Yeah. And Chris. All right. All in favor? Meeting's adjourned. Thank you very
Awesome.
much.
Thank you so much.
Thanks, Olga.
Good night, everyone.
Thank you, John.
Thanks, everyone.
Safe travels. Bye.
Beep, beep, beep. And Jim is zooming. Okay. You're good?
I'm good. You're good?
I'm good.
Awesome.
What do I got? That's bad. Okay.
I didn't mean. Oh, yeah. Okay. Sam, you're good.